So, how do you extract the soil? So, there are so many methods through which you can extract the nematodes from soil, but the most popular and most adapted method is cross decanting and sieving. So, you should know this method very from, I think this is practical thing. Through theory, you cannot get anything. But whenever you come in the online class, I will tell you with doing this. So I want to show you through figures. So this is the pan, different types of pan you can keep with you. And whenever you take soil samples or like compost, you know mushroom cultivation? Hello? Just respond me when I ask. You know mushroom cultivation, the white button mushroom. Have you eaten white button mushroom?
No, ma'am.
No, ma'am.
You don't know white button mushroom? Mushroom you haven't eaten any time.
Yes, ma'am.
That button mushroom is cultivated on compost. Okay, there is different type of ratios of different components. So in that case, also nematode attack is there. That is myceliophagous nematodes. Myceliophagous means fungus-eating nematodes. So as you know, mushroom is also a fungus. So here also the mushroom nematode infection is there. So for that you have to extract the compost material. So here you are seeing this compost material is what. So if you bring soil and whether it is compost, whether it is soil, you have to first keep in a pan with water so that all the clots should be broken. It should be a homogeneous mixture. But that mixture, when you collect soil, that soil should contain root materials and some stones, gravel, sands. So you have to extract it through 20 mesh sieve first to remove that part. Okay. So first you have to wet it in water and then you have to pass through it 20 mesh sieve. That time 20 mesh sieve is having very wide pores and through which you can remove the gravel, sand and that root materials. Okay. So that when you pass through that seal, you have to collect the extracts in another pan. That should not be extract. That should not be destroyed. So again, you have to pass through it directly to 325 mils or 500 mils, or you can pass through different mixes like 60 mils, then 100 mils, then 200 mils to get different size of nematodes. You know when maize, why it is called 100 maize seed, 200 maize seed? Can anybody tell?
Number of pores, number of C pores present per square inch.
Per square inch? No.
Mesh number.
100 maize seed means, if it is 100 maize seed, then it means in 1 linear inch. Okay, just remember, in 1 linear inch, there are 100 pores. Okay, So if it is 500 mils, that is the final. So that contains in non-linear range 500 pores. So that 500 pores in non-linear range means very fine pores. So if you can transfer this extract this after 20 minutes direct through 500, so that will block the pores. So you have to keep it patiently, and you have to turn the this. You can see in this picture, this seed is kept in some diagonal position. Okay, not in flat position. You have to keep it in some slopy position, and then with slight tap. So with one hand you keep the seed, and with slight tap you just pour very slowly water, that is soil solution. So with tapping we have to keep on the soil solution through this, and now you can discard the soil solution and the water which is coming out from this, because in this sieve all the nematodes are extracted, all the nematodes are sieved in this surface. So now you can discard the coming out of the soil and water. So again you have to wash the sieves content, because that is containing some soil material. So you have to keep the tap on, and the tap water should not be very fast, because if it will be very fast, then it will kill the nematodes. So you have to keep a hand on the water current, so that the water current should be very slow and very it should not be harsh on the nematodes. So just a slight tap you can wash, and when the fresh water will come, when the water will not be dirty, then you have to keep these sievings in a beaker. First you keep the sievings in a beaker, collect in a beaker, and then keep in this type of material. You have to, actually this is a modified Behrman funnel technique. You might have studied that my Behrman funnel technique, through which the funnel is used for sieving for extracting the nematodes, but that is not feasible for large samples. So this is the modification of that technique. So what we have done, so here you can take a petri dish. Here this is a plastic one. So you can take petri dish, and in which you keep the wire mesh. This is wire mesh, and it is a hard structure to support the tissue paper. So you just keep this wire mesh according to the size of petri dish, and put this facial tissue paper. In the tissue paper should be of very good quality, and this facial tissue paper should be in double folded, not in single folded. Otherwise, when you pour the soil sample, this extraction material, this will break, and after breaking, the soil material will pass through this tissue paper, and the extractant will be dirty, so we can't see the nematodes. So take care that this tissue paper should not be torn, and it should be wet. So with water, with hand, keep a small water on this and wet it, and fold this tissue paper from the sides. Why? Because if the tissue paper is keeping here, and if it is hanging around here, all water you are keeping here, that will flow through that to all the nematodes, because you know nematodes are hydrophilic. So whenever the water is there, wherever the water is there, it will go through that. So you should take care that this should be folded inside, and it should not be hanging. Now that water collected in that beaker, you should pour very slowly, keeping the layer of tissue paper, pour inside that, and cover it with another petri plate. Here we have some other structures, some other materials, plastic material is there. So discard the soil which is kept, which is left behind inside the beaker, because when you when you stir the soil with water, the nematodes are being lighter, they are floating in the water. And the soil particles, they settle very fast than the nematodes because soil particles are heavier than the nematodes. So during that period, you just pour that water with the nematodes and soil inside it, and the soil particles which are heavy, so left that one. Now keep it overnight and take the water that is coming outside. So this technique helps to clean the water because this tissue paper cleans the water. What happens? The soil particles left behind on the surface of this tissue paper, and water comes inside that tissue, inside that petri dish. And the nematodes which are present in this solution, they penetrate the tissue paper and come inside the petri dish. So what happens also don't go inside because some nematodes are in molting stage. Some nematodes during this process may get death. So dead nematodes, molting nematodes, molting nematodes are non-moving and non-feeding nematodes. So during molting, so these molting nematodes, some eggs may be there. So these all are present on this. So there is a different technique to extract this type of nematodes and eggs. We will discuss later. So all the motile nematodes, all the moving nematodes, they will come inside the petri dish. So next day, what we'll do, we'll collect those water and just leave behind just a small amount of water you leave behind, and by stirring, because some nematodes overnight they settle in the bottom. So when you stir that water with the soil with the petri plate surface, these all nematodes will come inside the water. So you have to take care at every step so that no nematodes will be left behind. So after that, what we'll do, we have to count. Okay. So now estimation of nematode population. Now you have the clear water with nematodes. So when you come, I will show you how to collect the nematodes, how to count the nematodes. So for counting nematodes, suppose you have taken, generally the standard size is 200 gram, or you can say 200 cc sample. So 200 cc means you keep the soil sample in 200 ml beaker. Okay, fill it up to 200 ml mark, and then you extract that amount of soil, and that is called 200 cc soil. So that 200 cc soil will give how much water. So after final extraction, fresh water, collect the clear water with nematodes. So measure it. First you measure it, and let's it come 50 ml. Okay, so 200 cc soil gives 50 ml extraction. That 50 ml extracted soil water sample with nematodes, we have to estimate the nematode population. So for that, we should have nematode suspension, counting this, and tally counter. So I will tell you, I'm telling you about the expression of nematode population when you get the nematode suspension to the hot sealing decanting method. So now you have to get pipette, measuring cylinder, glass slides, and stereo microscope. Okay, so in nematology, we work on two types of microscopes: stereo microscopes and uh, compound microscope. Okay, so have you studied about stereo and compound? Anyone? Hello, are you listening to me?
Yes, ma'am.
Do you know what is the difference between stereo and research microscope, research or compound?
Maybe stereo microscope is used for low magnification, or compound microscope is used for high magnification.
Have you seen how they are? Just so this is the stereo microscope. You see, okay. So many different types of models are there. So here it is binocular stereoscopic microscope. You know, in the stereoscopic microscope, you can work. So working distance is more. Here you can say here, from here to here you can work. So this is a stereoscopic microscope. Here the magnification is less, up to 65 or 100, and this object, this eyepiece will get 10x. 10x means 10 times magnification is there, and here it is 65 to 100. So up to 10 into 66.5, or 100 means 10 into 10. So total 100 times magnification is there. Okay. So this is called stereoscopic microscope or working microscope. And this is your research microscope, or you can call it compound microscope. So here when you prepare slides, so here you can keep your slides, and these are having 4 to 5 more objective lenses. Okay. So it starts with 4x, then 10x, then 40x, then 100x, sometimes 60x, sometimes different microscopes are different objectives, but mostly 4x, 10x, 40x, and 100x are there. So with 4x, 10x, and 40x, you can see directly, but for 100x, you have to use immersion oil because that magnification is very high. This eyepieces have 10x, and this is 100x. So you know it is becoming 1000 times magnified structures. So for that you have to reduce the refractive index, and for that you have to use the immersion oil. So one drop of immersion oil, and that after that you can see. Otherwise you cannot see through 100x. So here you can see the working distance is very less. We have to attach these lenses with the slide, and then you can see through this. So this is the basic difference. So counting will be done here, and for counting you should use counting dish. Okay, so this is hand tally counting. You just, this is a loop, you can keep inside the thumb, and with this you can remove these digits. So here 4 digits are there. So keep it at 0 and then count it. So whenever this is a tab structure, so when you keep the tab, if you see one nematode, keep one tab, it will come 1. So you can count the nematodes here. So here only you can count the whole nematodes, and if you know whose if you identify the nematode, then this tally counter is used. Here you can see different tabs are there, and this is the knob through which you can make the point 0. All the digits are 0 before the counting starts. So when you count, like if you know that this is Melodogam nematode, this is cyst nematode, this is Tylencorhynchus, then you should write here. So you might see here that this is like written T, then H. H means Hoplolaemus, and T is Tylencorhynchus. HE is Helicotylynchus. So if you know the nematode, then this tally counter is useful. So whenever you know that if it is Helicotylynchus, keep one tab here, then it will come 1, and here it is total number. So here 1. If you see Tylencorhynchus, here keep 1 or 2. If you see 2 nematodes of Tylencorhynchus species, so tap 2 times, it will read 2, Then 2 plus 1, the total will be here 3. Okay, so when you identify, this will be helpful. But if you don't know the nematode identification, this hand tally counter you should use. And this is your counting dish. So this is made of plastics, and here you can see there are marking of equal structures, equal squares is there, and keep your water here. So first you have to blow the suspension with pipettes. Okay, so these are the different types of sieves. You can see here this is 20 ml sieve, then 60 ml, then 100 ml, then it is 500 ml. Then these are different types. So these two are called picker. Picker means, you know, here very sharp tip are there, and you should make it under microscope, under stereos microscope. This is a peacock feather. This is made from peacock feather. So you can use that nariyal broom, or you can use any that crow's feather, that midrib of crow feather you can use and sharp it for picking the nematodes. So it should be of thickness of nematode body. Otherwise, you cannot pick it. This is the forceps, and this is brush, and this is needle. So with needle, we can puncture the root knots, and for picking, you need this one. Okay, so what we'll do after that, we have to count the nematodes. So how do you count? So first you should take 5 ml or 2 ml pipettes. I was searching for pipettes for pictures, but it's not there here. So the pipette, you know, it's a rounded cylindrical type structure having hollow structure. So when you blow it through the soil and that nematode suspension, it should be homogeneous. You know, the nematodes after some time will settle down at the bottom of the beaker. So you have to first get it homogeneous solution. So for that you have to blow it with pipettes. So you have to blow it so with the all the nematodes will be in nematode suspension, and that will float. So that time you should suck 5 ml or 2 ml according to the need, according to your need. So take 3 times, at least 3 times. In that counting disk, keep inside that liquid and keep in under the stereoscopic microscope and start with the counting. So how do you count? So if the water covers like 6 squares or 7 squares or 8 squares, so first start with the top layer. So let's see, let's suppose that you start from here. So you go from here to here or from here to here. So one style you should follow. So if you count, so this first square should be in focus in under microscope. So you should count how many nematodes are there in one square. And then if you come from here to here or from here to here, then you should remember that you are going from left to right or from upper part to the lower part. So if you go from left to right, then again the second square is come. Second square comes, then you have to count the nematodes from second square. then to third, then to fifth, sixth, and to last. So when you come to the last, then you go from upper to lower part. So in second line, if you come from second line, then you have to go from right to left. So in this way, you will not leave any square, any part. So you can count all the nematodes from the liquid you have poured here. So up to the last point, you should count all the nematodes, and that count you should write in your paper. So in 3 counts. So 3 times you count. Let's see, in one count you counted 8 nematodes, then in second count 10 nematodes, in third count 12 nematodes. Then you have to take the average of all 3 counts. Okay, suppose you are taking 2 ml in 3 times, then it will be 10 plus 8 plus 12 by 3. And suppose it will come, how much it will be? 30. So if it will come, the average count is 10 nematodes per 2 ml. So you have to calculate for 1 ml. So in 1 ml, it will come 5 nematodes, and your total solution is 50 ml. So multiply it with 50 ml. So 1 ml is having 5 nematodes, then 50 ml is having 50 multiplied by 5, 250 nematodes. So what does it mean? You have taken 200 cc soil and you are getting 50 ml nematode suspension, and finally you have calculated that 250 nematodes are there. So this count will tell you that 250 nematodes are present in 200 cc soil. So this is the nematode estimation. So if you further identify the nematodes which are plant pathogenic, which are not plant parasite, which are beneficial nematodes, then you can say that this soil sample contains this much of plant parasitic nematodes. So before planting a crop, before sowing any seed, you have to go for control measures, or there is no need of control measures. So according to the identification of these nematodes. So what is the peculiar characteristic of plant parasitic nematodes? Anybody can say? We have